Ooh, recording is in progress. It will be speaking to us today about models of peptide hydrogels. Did I get that right? I think I got
Yeah,
that
yeah,
right. So I might just pass over to you, and we're looking
yeah.
forward to
Okay,
hearing
thank you
from
very
you.
much. Thank you, Joe. So to make sure can you see my slides now? Or okay, good. Okay, so thanks for the introduction. As Joe mentioned, I'm Amir. I'm a PhD student at the University of Manchester. I work on modeling of peptide hydrogels. And the title of the talk that I'm going to give today is the study of percolation and mechanics of stochastic fiber networks as a model of peptide hydrogels. And obviously, I'm first gonna start with the motivation, why I'm doing the stuff that I'm doing. After that, I'm gonna describe my network model. After that, I'm going to talk about how I create a network, which is the modification of the classical Mikado network. After that, I'm going to use that method to investigate behavior of the system at low and high densities. And at final slides, I'm going to compare my result with some experiments qualitatively. So I think main motivation of doing this investigation is many materials in nature and man-made materials are made of fibers. They connect to each other. They form a big network of fibers in space. We can see some natural examples on left, and we can see some man-made examples on right. Therefore, and there are plenty of different kinds around us. So obviously, it's very important to understand how they work. And because obviously it's gonna make us more capable of dealing with this sort of materials. The specific material that I'm working with, or I collaborate with some experimentalists that work on the field, is peptide hydrogels. So the peptide hydrogels, as the name suggests, is formed by peptides, which are a sequence, like a short sequence of some amino acids connecting to each other, anything between 4 to 20 amino acids connecting to each other. They are forming a chain called peptides, and which we can see here. And these peptides, if the condition is suitable, they can connect to each other. They form a fiber. And this fiber, if the density is bigger than a threshold, they can create a network and form a solid. And since these hydrogens are made of amino acids, they are very biocompatible. People use that for bioengineering, tissue engineering. I think there are many people in the laboratory that I'm collaborating with to test different sort of, they do cell culturing, they test different drugs on the cells to see how they're gonna behave. And something that's very important here is that the mechanical behavior of this structure of this hydrogen should be similar to our real body, to extracellular of our body, to matrix of our body, to make sure that the cell is gonna behave the same, because otherwise the behavior of the cell would be different and their reaction to drug would be different as well. So here is like some example of different peptide networks we can see on top and bottom. The only difference is the last chain of amino acids, last two chain of amino acids, the last two blue ones. And we can see the network that they form, they look very different and they behave mechanically very different as well. So it's very interesting to investigate how this happens, by it happens, and how it affects the mechanical properties, which is basically my work. If you go to the previous picture, we can see the structure of a typical structure of a single fiber on bottom. We can see on the edges they have a hydrophilic surfaces with, and on the center we have a hydrophobic core. The hydrophobic core tends to bring two peptides together and form a sheet that we see here. And the other hydrophilic surfaces bring many of these ones to make like a Lego-like structure and form like a very long fiber that we see on top. And by changing these amino acids, because for example, the charge groups on the sides gonna be different, they're gonna have different properties as well. So for example, they can attract each other more, or they can bring like a different form of interactions, and they want to like push each other away. And it's kind of very easy to control them. And since there are many different type of amino acids, we can make like engineer that in many various ways. So my project is basically to investigate the structure that might rise with these networks, and how this structure is created is something I don't care about. Is I get that from experimentalists. I just have different network structure and investigate how that might affect the behavior. So just imagine the general idea of the work would be, so we first generate some network like that. We define any connection. It can be anything. It can be they can be basically welded together, or they can be cross links, or uh, any different time is also possible. After that, we deform the structure as we see on the right. So for example, we can apply a shear deformation or a stretch and see how the network deforms, how energy absorbs in the network, and based on that, we can investigate various mechanical properties like Poisson ratio or modular or shear modulus or UI modulus of the network, and see how they're gonna change. If I want to show how the network is also created in the model, so basically we add a sticks of length L into a square box of size of like big L, and then we investigate the connections. And if we have a fully connected networks from top and bottom, after that we can apply boundary condition to see how the network behaves. And can I go to next page? Okay. So as we add fibers in the space, we can see different stages occurs. The first stage is that we don't have a percolated system. We have like a smaller batches that are connected together. We basically don't have a solid phase here. And it's basically equivalent to say when we want to create a actual hydrogel, we haven't added enough material to a network. If we keep adding materials, at some points the network's gonna percolate. The correlation length is also defined as the average size of each cluster that we see here. Before percolation and after percolation, basically going to be the average size of the cluster, except for the infinite one, except for the percolated one. And after a while, we're basically going to end up with just one fully connected network, which we're probably going to see this one more in the real application. People care about this one more. And I'm going to refer to this one as high density networks, and the other one, like just after percolation, as the low density networks. So the first thing, obviously, is to investigate the percolation of the networks. If we have a system that its size is infinity, we can say percolation either, so there's a percolation threshold before which percolation doesn't happen, and after that, percolation definitely gonna happen. So the probability of seeing a percolation gonna be either 0 or 1. But we cannot obviously model an infinite size network, so we have like a smaller size with size of capital L. In that case, we can see if the density is low, the chances, the percolation probability is going to be low. And as we increase the density, it's going to increase as well. And due to duality, we can say at some point, at the density equal to density of percolation, the probability of observing the percolation at each system size is going to be basically half. And if we do further investigation, we can do basically, we can say at density equal to density of percolation, there's a function that is follows, and that function is it depends on the system size that we have, and and we can basically introduce that by dividing that into how the cluster size differs. There's going to be function. We don't know how the function going to look like, but since we are interested in very small area, we can basically say we can approximate that by Taylor series. And since it's the odd function, we get rid of all the even terms, and we end up with this thing with some small extra step. And it basically can end up with this function on the bottom here, which basically says if we observe the percolation probability equal to 50% at different system sizes, we can basically extrapolate that to infinity and investigate and find out what's the percolation density that we are interested in, the percolation density that happens at the system size of infinity. So that's going to be the first thing that we're going to investigate. After that, we're going to be investigating the mechanical behavior. And mechanical behavior is going to be divided into two sections, as I mentioned, near percolation and at higher densities, which the system is fully developed. At near percolation, we basically have a phase transition somehow. During that area, the change of the elastic modulus, people have shown that it's going to be exponential function of this form. Oh, sorry. Of this form, density minus density of percolation in power of f. And we're going to use a scaling method to investigate the mechanism during that area. So, and for the fully developed networks, the investigation going to be slightly easier because we just need a representative volume element to do our investigations. And during that area is people have shown that our investigation can be divided into two sections basically, where initially bending is the major form of absorbing energy, and after we increase the density, the stretching gonna be more dominant way of absorbing energy. And during each of these, how the modulus changes with density basically is they behave different. At bending dominated regime, the modulus is gonna be influenced by a function like that. It's gonna be exponential with a power of fh. And at higher density, which fh is gonna be dominated, it's almost gonna be a linear function of density. So in the classical way of creating network is that we just randomly put network in a space, and the orientation also going to be randomly allocated. But since we wanted to investigate the microstructure and see how the microstructure going to affect the network, is it going to be effective or no at all? So we modified the way we create the network. So we said, okay, we first modify the dispersion of this network. So we want our networks to basically disperse in space more homogeneously. And to do so, we allocate a chance around each already existed fiber, which we basically reduce the chance of appearing a new fiber in that area. So what we do whenever we want to add a fiber, we look at its surroundings, we count how many fibers already exist there, and based on how many fibers exist, we reduce the chance. Therefore, the chance of appearing a new fiber in the areas that there is no fiber going to be higher. Therefore, we expect the network to be more homogeneous. In the dispersion and next step, we also modified the orientation of the networks as well. So after placing each fiber, we need to allocate an orientation to that as well. And to do so, we look at its surroundings, and if there is a fiber close enough to the fiber that we want to add, we get the orientation of the fiber from a Mitchell-Gaussian distribution. And for example, if we want our fibers to be perpendicular or parallel to each other, we can modify that by changing this capital theta here. An example of like each fiber, each basically network can be seen here. The first one is the classic Mikado network. This one is something that people used to develop the investigation. It's somewhat well understood in the literature. And the spatial modified network, an example is here. As you can see, the network is kind of more homogeneously dispersed in space. And for DLM type 1 and 2, which we can say here, I wanted the network to be more perpendicular to each other. And for the DLM type 3 and 4, I wanted the fibers of the networks to be more parallel to each other. So we can see different batches in different places that fibers are basically kind of parallel to each other. And these are basically the parameters I used to create these 6 types of networks. So I wanted to investigate, so does it going to be effective or not? And okay, so the first thing after creating the network was to investigate the percolation. I used the formula I explained earlier. I did simulation at different system sizes, and then I extrapolated to when the size of the system going to be equal to infinity, and I investigated the percolation threshold. So for the Mikado network, 5.63 is something already existed in the literature. So using this method, I can end up with that value. And I use this method to investigate the new types as well. And we can see the percolation threshold change dramatically from 4.1, the smallest I had, to 9.4, the largest I had. It kind of showed that if system have tendency to be perpendicular. The fibers want to be perpendicular to each other. System percolates much faster than when the system wants to be parallel to each other. It's kind of an observation we had. So other features of the networks that are important in the mechanical behavior of networks are also investigated here. In the first one, we divided the system size into 400 smaller boxes and count how many fibers exist in each box. For a fully random process, it should be Poisson distributed, which means the mean and variance should be equal to each other. So mean and variance for when PR is 0, similar to each other as expected. And when we increase the PR, which we expected the network to be more dispersed in space, we can see the variance is decreasing, and the networks got more homogeneous. This set is the connectivity of the system. So basically, it says each node in our network connected to how many other nodes. This is the average number on that for the whole network, for the full networks. And by the way, all the results here are for after calculation. We can see this value is somewhat similar for the first 4 type. So they are slightly different, but they're very close. But for the last 2, it's still very close, but the difference is more pronounced. And the last 3, I need to remind that is for the system that the fibers are parallel to each other. This one is the average length of segments, and segments is the area of each fiber that are between 2 consecutive nodes. As you increase the density, this value definitely should decrease. And again, first 4 networks are somewhat similar, but the other 2 are kind of more different to the others. So the average size of the segments that we see gonna be larger. It's kind of important in the sense that the transition from bending to stretching gonna happen at a later time. In this time is basically we calculate how many is the average density of segments in area, and we can see the last to the perpendicular one. The parallel ones have like the less of this value, and the others are somewhat similar to each other. So as I mentioned, for near percolation, we use scaling to investigate their mechanical properties. As a reminder, the modulus will change by a function like that, density minus density of percolation in power of an exponent value f here. So investigating that is very difficult in the sense that we cannot make an RVE because the cluster size just after percolation is huge and we cannot computationally create them. So what people have done, they basically proposed a function and they said, okay, so we know, like, because we are doing our investigation smaller than the cluster size, so the change should be a function of size of the box divided by this value of cluster size. And since the cluster size changed by this function here, basically can create this function h, multiply that by this value that we have here. So near percolation, this or system should follow the scaling function. So what we need to do here is to just do some simulation at different system size, different densities, and class all the data to form a function as such. And based on that, we can find the value of f. Basically, we change f until we form the function h. And the value of f is 3.77 for the Mikado network, and it changes for other network types. The change might not look very big here. It's kind of between 3.5 to 3.9. But I think, and for many systems, it's actually not important. For one system that's actually very important is colloidal gels, because this system, they naturally have kind of a fractal shape in their fibers because they basically some fractals connecting to each other randomly. Uh, if we consider two nodes in the system, the edge that they form between these nodes, it kind of looks fractal. And people proposed methods to investigate them based on this value of f. And for those networks, since the value of f, we've shown that interaction can change this value of f. People should basically consider this value because previously people already considered this constant for the one that gets from the Mikado and apply that to every system. It changes slightly, but for different systems, in reality, this difference might be much, much higher, considering how intense is the interaction of the colloidals. At a higher density, when we are in the bending dominated regime, so the modulus change will change by density in power with power law of the exponent going to be f. I call that fh here, f high. And when we are going to a stretch limited regime, it's going to be basically going to be a flat plateau, and that's going to basically going to be a linear function of density. In the bending limited regime, which means we calculate how much energy is absorbed in bending the formation of each fiber and how much energy is absorbed in stretching. If the bending basically basically if we consider all energy of the system and bending basically contributes to 99% of that and more than that, we define that as bending dominated regime, which here it should be somewhere from here and below that, that's lower densities. You can find the value of FH can be also different. The Mikado network can be 7.9, similar to what's already reported in literature as 8. For the DM and other DM types, it's also going to be different. And for more parallel networks, which we kind of expect to see more heterogeneity, we can see this value is going to be higher than 8, higher than others. And we already saw in previously when we were investigating the structure that the structural properties are kind of more similar for the first four. So it was kind of expected to see that mechanically they're going to behave the same as well. Something that we observed and reported kind of for the first time was how the Poisson ratio changes in different networks. So belief was that the Poisson ratio only depends on which regime that we are at. So either we are in a bending dominated regime or a dominated regime. But we have shown that actually if we are still in the bending dominated or SH dominated and we change the density for some reason, Poisson ratio is also changes. It's not constant. So what we have here is the Poisson ratio with respect to density for conditions that we can say we are in a bending dominated regime. It's not more than 99%, but it's more than 90%. So we are still bending is more dominated. It's expected to be constant, but it's not. And to investigate the reason uh, for that, we should basically look at the structure and how the energy is absorbed in the network. So from these two pictures, we basically on top right, we have two networks with different densities. And and the red ones are basically absorbing 50% of the energy, like the fibers that are absorbing the most energy, basically. And the blue ones, I think uh, they are involved in absorbing 10% of the energy, but the fewest, so basically it means the highest number of fiber that we can find, the highest number of sets that absorb 10% of the energy. And the red ones are the fewest number of fibers that we can find that absorb 50% of the energy. And as we can see, although it's probably not very clear, here, let me just zoom in. So it would be much. But at the average size of between these two, the average size gonna be different. So let me just show that one here. So this is basically the average segment size that absorbs the 50% of the energy. Initially, at lower density, this tends to be larger than the average size of the segments. But as we increase the density, this value decreases. So, but we can deduct is that in this condition, we have basically some higher density areas connected by, in the higher density area, the average size of segments are lower. But these high density areas are connected by less dense areas, which the size of the segments are kind of higher. In the first one, these low density areas absorb most of the energy. But as we increase the density, the story changes. And the high dense area basically absorb the energy, which kind of explains, and that's kind of shows why the deformation structure, despite the fact that we are still in the bending diameter regime, changes. And that's and since the deformation structure changes, the Poisson ratio also changes, decreases or increases. This one is also this realization density, and in this one, the omega here is the how much each node rotates in space, and we can see at lower density and higher density, the distribution changes a lot. And at higher density, basically, many more nodes are involved in rotation, and basically somehow shows like more areas are involved in rotation. So those high dense areas that I mentioned, initially they used to just deform and move around and do not rotate a lot. But as we increase the density, they become more involved in the formation, and the rotation also occurs over there as well. And we can see this value, how it basically, how the average of that basically changes by increasing the density. Obviously, absolute average. And so as I mentioned, to create different structure, it's what people do if they change one amino acids, the interaction with other fibers and water gonna be different, and therefore they can form a different structure. But people in the experimentalists to form like different structure D, but that they change the last sequence of amino acid chain. This is F. It's I forgot its scientific name, but it means this is a hydrophobic core. So they want to create more interaction with other fibers because obviously they are hydrophobic. But this is a hydrophilic amino acids. Therefore, it wants to be nearby other of water and not other fibers. So we kind of expect that this one gonna be more similar to uh, perpendicular networks, that the connection was lower than the others. And this one to look the first four, since the connection was more than other network types. And in the last one, we added a hydrophobic first, and then a hydrophobic first, and then a hydrophilic. And the action, since basically the interaction is basically happens by the last one, we can see that in our literature, these two basically behave similar to each other. But since this one has one more amino acid in the chain and has like more rigid fibers, it's just the value that we observe for the shear modulus is slightly higher, but the behavior is somewhat similar. Also, we can, and we have some TM picture here, but we cannot deduct much from these pictures on the structure. And the reason is that to get the picture, they need to basically form a two-dimensional network. They get the water out, they dry that on a sheet, and they take a picture on the sheet. And therefore, kind of the information on the structure get lost during the area. But what we can observe here is that the thickness of each fiber are almost similar. All the peptides that we use form basically the network, because all of these can be effective in the behavior of the network. And we cannot see any uh, clear reason why these three networks are different, especially is that why their exponents are different. But by what we showed in our modeling is that if we change the structure even slightly, we can see that behavior is going to be different a lot. Like also the exponents of this one F is a smaller 1.6, but it has the higher value. It's exactly similar to what is observed in our experiments. On the other hand, this one, which kind of more similar to the perpendicular networks, we can see that they have a higher exponents, but lower shear modulus, which I think is similar to what we observed. So basically, our investigation suggests a possible reason for this, because all the other possible reasons can be crossed out by looking at these PM pictures and how similar the structure of these peptides are. I think that's it for today. Thanks for attention, and apologies again for me having a weird problem, kind of make me feel very nervous as well. And so my talk today was based on this paper, which is I got the acceptance last week, and if people are interested to reading that, they can just find that right now in archive. Thank you.
Thank you very much. Has anyone got any questions for Amir with that?
Okay. Your model, so how specific is it to your peptide hydrogens? Like you're doing this percolation. It seemed pretty general to me. Is there anything specific about the all these results? Are they effectively all you're setting is some rules for how you put your fibers in, right?
Yeah. So basically, just I think the math behind that was already developed. There isn't anything new on the math side of it, to be honest. I think the result was the novelty of our work, and we just wanted to investigate what is the percolation threshold, because based on that we have like a rough idea on what density we are basically in a high density area. So because in experiments, it's very almost impossible to observe the percolation experimentally because the network is so big, you cannot examine it with anything available. But we needed to investigate that because we needed to have a like exact value. First of all, to see how the network behaves, and second of all, to know how far we should be from this area to make sure that our investigation basically is not affected by the percolation. Also, experimentally, an observation that we didn't report as no paper is that in the experiments, the networks do not show like the solidity at the same density. And so, for example, the KF bond, this one, showed we needed to use basically kind of more denser, kind of more peptides, a higher concentration of peptides to form the gel, which is kind of equivalent to what we see from the percolation, but we didn't report that because that wasn't basically the main reason or main idea of our work.
Yeah. But just check. This might be a slightly silly question. But, what is your density?
Okay, so density is the number of density can be defined in many other ways, but basically our approach to the same thing is my density. My density is the number of fiber that I use, and the size of fibers are similar. So at lower density, and one other thing that people do is they investigate the lengths that are active, like the lengths that between different nodes. If you are at very high density, these are gonna approach together, but at low density, it's gonna be slightly different.
Suppose what I
But
want to know is what does like
uh,
density equals 1 or 1/2 mean? Like,
okay, so the number of fibers in space, in a square box by like 1 to 1. So so for example here, uh, just imagine I count how many fibers I use to Katie and divide that by the size of the box, and that's going to be my density.
okay, so the thickness doesn't matter. They're literally
Thickness
1D.
doesn't matter.
It's just compared to that size.
Yeah,
Thank
it's
you.
just basically the length.
Yeah. Okay.
I was going to ask something that might be a really silly question about this orientation parameter, this theta. Um, and you see obviously quite different behavior for theta is 0, theta is pi by 2 in the two models you've got. Where am I right in saying that that's where fibers either want to align perpendicular or want to align parallel?
Yeah.
Have more. How does that correspond to the network structures that well, the actual chemicals that you're talking about here? Are there
Uh,
certain polymer
that's
structures where they do want to align perpendicular or where they do want to align parallel?
yeah, that's actually a very good question. So what we have here is just math, and I don't think it can be like directly be connected to the actual network because we can see what happens there as well. But since in the, I mean, let's just look at this one first. So we can see here that the average size is kind of higher. The Z is the connectivity is lower. So it means it creates less connection with other fibers. And in the experiment, since we have used this amino acid F, which is hydrophobic, we expect that to have more tendency to make connection to
End
other
on.
fibers.
Yeah.
And this one wants to create less connection with other fibers. And that kind of happens if you want to look at that, think about that geometrically. It's like if they want to evade each other, they kind of have to be parallel.
Parallel.
And if they want to make connection, they need to be perpendicular in two-dimensional space.
I
And
suppose
someone kind of like that.
a
Yeah.
follow-up sort of mathematical question, and again, this really is just speaking about the maths as opposed to the polymer chemistry, which I know nothing about. But um, is that very big jump in differences in behavior between theta is zero, theta is pi by two? Is that continuous across different values of theta, or if theta's just either side of pi by four, do you get this
I,
sudden?
I, I would imagine it would be continuous, but I
Okay,
haven't investigated them.
yeah, that was a bit of a
Yeah.
random add-on there,
Yeah,
but
because I think my simulation, I needed to do many observations, and each of them takes so much time, so I
yeah.
basically needed to not do it.
Thank you.
Maybe I can ask a more experimentally focused question. Um, the models that you've simulated, the images you show at the end in the TMs, do these map onto your even simulations of those structures like the Mikado, etc.? You know, do you even
Um,
qualitatively, or could you quantify that in any way? You know, how
and
self-similar are those structures?
yeah, that's actually a very good question. Unfortunately, no. And the reason is how we create the image. We had so much conversation with the experimentalists. They mentioned it's going to be, if they want to, first of all, it's a three-dimensional network. It's a real material, it's a dimension. Getting the topology of that and how the networks connect to each other is very experimentally difficult, almost impossible. And if they want to do that for a very, very small network, it's going to be extremely expensive as well. So it's not doable. So what they do, they take a small amount of hydrogels, they dilute that 10 times, and then they put that on a sheet, which I don't know what it's called, and then take the water out. They basically dry that.
Mm.
And then they take the picture using KEM. So I imagine during this process, so much stuff happens that the network do not exactly, the structure of the network is basically lost. We cannot get that much information. Like we can get very useful information though. We can get like the size of the uh, networks, the fibers are similar, and aggregation do not are carrier, which aggregation has a massive effect on the behavior. We can see that it's not the case here. We can see that basically almost all despite the fact that we diluted this, we can see that all the material that we use basically participated in creating the fibers. So we can basically make a one kind of to one analogy between our density and the concentration here. Because otherwise we couldn't basically say how much of the material is formed in creating fibers, therefore investigating the effects of concentration would be meaningless basically. So we can see that's not the case here, hopefully. And
I still think here these look like entangled networks, even though you're not necessarily at representative concentrations.
yeah.
Could you simulate less dense fight? I mean, potentially longer, but something that could give you comparative simulations to these actual experimental conditions. Maybe not
Uh,
the mechanical properties might be less important there, but at least from a sort of network structure perspective, you know, do these
uh, okay. That's actually a good question.
aggregate or the correlation length in that sort of mesh size of which, if you're thinking of something like drug delivery, that might be relevant to sort of solutes or
That's
particles sort of transporting through these networks.
that's actually a good question. So I mean, I so when I was thinking what method should I use in creating my network, I have two main options. Use some statistical approach and create something, some stochastic process as I use, or I can try to insert some information on the base on the interaction that exists with fiber, and try to like create some fibers in a space, and by the interaction that exists, I do some simulations and see how the fibers interact with each other and how they form a network and a structure, and that probably would be more similar to the reality. This was another option as well, but that's kind of we made the decision that this might not be too much for the PhD, so we kind of gone we crossed out this approach and we kind of focused on taking the stochastic approach because it kind of gave us some idea of what's happening anyway.
Yeah.
And there was no guarantee that that approach going to work. So it kind of.
Very true. Thank you. Thank you for the lovely talk, by the way. I
Thank you
should have
very much.
led with that.
Thank you.
Take the silence to mean you've answered all of our questions.
Okay.
Thank you so much for
Thank you
dropping
again.
in amidst an incredibly panicked kind of
I
tech
know.
setup. So thank you so much for delivering the talk and for stepping in.
Thank you for having me. Too. Thank you.
Yeah,
It was an
and
honor.
we'll see you around, I'm sure.
Yeah, see you, sure.
Thank
See
you.
you.
Thanks.
See ya.
Bye.
Bye bye.
Bye.